Hello, class 10. Today I am resuming the chapter of circulatory system from life and death of RBCs. In adults, the RBCs are produced in the marrow of long bones, especially in the ribs, breastbone, and ileum of hip girdle. In an embryo, they are produced in the liver and spleen. In children, they are produced in bone marrow of all bones until 5 years of age. The mature red blood cells have no nuclei. The average lifespan of an RBC is about 120 days. The old and weak red blood cells are destroyed in the spleen, liver, and bone marrow. In a normal adult, approximately 2 million RBCs are destroyed every second. These are some of the points which you need to learn or remember rather in order to answer the short questions. You may get it in your MCQ portion. Okay, and very important point among all these are the two. First of all, which mentions that it has no nuclei. This is one of the very important points among all these points. And another one is about the lifespan of RBC, which is about 120 days, and you have to remember this. Okay. Now this portion is also a very important part. Deficient but more efficient. Mammalian red blood cells, when mature, circulate in the blood system and are devoid of certain organelles. They have no nucleus, no mitochondria, no endoplasmic reticulum. That means they are deficient of all these organelles. Though deficient of the above organelles, the mature red blood cells in reality are more efficient in carrying out their task of picking up and delivering oxygen. The factors making them more efficient in these work are as follows. Loss of nucleus makes the red cells biconcave, thus increasing their surface area volume ratio for absorbing more oxygen. As you can see the diagram they have given, how without nucleus it is helping the RBCs to become biconcave and increasing the surface area volume ratio. Space in between increased, more RBCs can be accommodated in the same space. Loss of mitochondria means that the red cells cannot use oxygen for themselves, thus all the oxygen absorbed from the lungs is transported and delivered to the tissues unconsumed. And the third one, no endoplasmic reticulum, means increased flexibility of RBCs, because endoplasmic reticulum gives a very strong mechanical framework, and absence of this means increased flexibility for their movement through narrow capillaries. Okay, so this is a very important question in give reasons, and I hope you can give it because they have given very nicely in the book point wise. So deficiency of three organelles, how they are helping them out in absorption and carrying of oxygen, that you can describe. Okay, now another very important thing which you have to remember is abnormally increased number of RBCs is called polycythemia, and their abnormally decreased number is known as erythropenia. Abnormally increased number of RBCs, that means poly means more than 2, so polycythemia, and their abnormally decreased number is erythropenia. These two terms you have to remember. Now next cellular element is WBC or leukocytes, that differ from RBC in having a nucleus and not containing hemoglobin. The number is much less, 4,000 to 8,000 per cubic millimeter of blood. Most WBCs are amoeboid and can produce pseudopodia, with which they can squeeze through the walls of the capillaries into the tissues. We call the phenomenon as diapedisis. This is again a very important term which you have to remember along with the diagram figure 8.3. Please practice the diagram. Based on shape and other characteristics, the white blood cells are classified into two major categories, granular and non-granular. Now you see just beside this page they have given this table. This table is very important because here they have mentioned properly the granular and non-granular classification and what are the five cell types among these granular and non-granular types, what are their appearances, what are their distinguishing features, and also their functions and site of production. Function is very, very important, students, so please go through this chart. This chart is very important chart. Now let's come to origin and life of WBCs. The WBCs are produced in red bone marrow, lymph nodes, and sometimes even in liver and the spleen. Their average life is about 2 weeks. The neutrophils live for only a few hours, and about 125 billion neutrophils are produced each day. The old and worn out WBCs are destroyed in the same manner as the RBCs. Leukemia is a cancer of the tissue forming WBCs whose number increases manyfold at the cost of RBCs. It is usually a fatal disease. Currently, the treatment is only blood transfusion. This is actually, we all call it as blood cancer. It is actually leukemia. Okay. Leukopenia is the abnormal decrease in the number of WBC. Just the, like RBC excess deficiency, that means erythropenia, and abnormal decrease in number of WBCs means leukopenia. Okay. Now, functions of leukocytes or WBCs. Phagocytosis is one of the very important function because just now we have read that they are amoeboid and they can uh, bring out their pseudopodia in order to pass through capillaries, very fine capillaries. An abnormal increase in WBC count up to about 50,000 or more per cubic millimeter indicates some infection in the body. Inflammation occurs due to the reaction of tissues to injury and to localized invasion of germs. The inflamed spot has several characteristics: increased local heat, redness, swelling, pain. Here, the leukocytes migrate through the walls of the blood vessels by diapedicys and fight against the disease-causing germs. They also destroy the damaged cells by phagocytosis. Now, what is pus? Pus, the white thing which comes out from the injured part. You all know that. Pus is actually composed of the dead white blood cells together with the tissue cells destroyed by the bacteria. Formation of antibodies. The WBCs produce antibodies which kill or neutralize the germs or the poisons which is coming from them. Now, introducing weakened germs or germ substances, we call them as vaccines, during vaccination stimulates formation of particular antibodies which at a later period would destroy the particular disease-causing germs if they enter into the body. So, formation of antibodies is a very, very important task performed by the WBCs. Okay. Next cellular element is blood platelets. Blood platelets are the initiators of clotting of blood. We also call them as thrombocytes. They are very small structures, non-nucleated, and platelets are derived from some giant cells called megakaryocytes. Very, very important point, students, in the red bone marrow. These are budded off from the megakaryocytes in a manner that each one is completely surrounded with membrane. Their lifespan is very short, 3 to 5 days, and are destroyed mainly in the spleen. Spleen, it's a lymphatic organ. They are very important in clotting of blood. At the site of injury, the platelets disintegrate to release a chemical substance, thrombokinase, which initiates the process of clotting of blood. Now, we have to know how this clotting of blood occurs. Okay, come to the next part of this page. First of all, explanation is very important, and then you can see just beside that main reactions in the blood clotting they have given in a form of flow chart. This flow chart is also very important, which you have to practice. Now you see what they have written about the explanation. Now, when a blood vessel is cut, blood escapes from it, we all know that, but soon a clot is formed and the flow of blood is stopped. Clotting occurs in a series of steps. It's not such an easy process, okay? So it takes a lot of things to happen. The injured tissue cells and the platelets which disintegrate at the site of the wound release a substance thrombokinase. The thrombokinase acts as an enzyme, and with the help of the calcium ions present in the plasma, it converts the inactive prothrombin into thrombin. Now, thrombin in the presence of calcium ions reacts with the soluble fibrinogen of the plasma to convert it into insoluble fibrin. Fibrin is a solid substance that forms threads. These microscopic threads of fibrin are sticky and form a network at the wound. Blood cells are trapped in the network of the fibrin. The network then shrinks and squeezes out the rest of the plasma, which is in the form of a clear liquid, the serum. The solid mass which is left behind is called clot. So first thing which is being released is thrombokinase, and this thrombokinase is acting as an enzyme which is not itself taking part in the reaction, but it is helping out the reaction process, like it is converting prothrombin to thrombin with the help of calcium ions. Again, this thrombin is acting on fibrinogen to convert it into fibrin with the help of calcium ions, and ultimately this fibrin is forming a mesh of network sticky threads which we call as clot. This main reactions in blood clotting, this flow chart is very important. Okay students, today up to this, please go through the chapter till now, whatever I have taught regarding this chapter. If you have any problem, come up in the comment box with your problem and I'll definitely solve it. And I hope the previous portion of this chapter was also very clear. Please go through it and I'll come to the next part of the chapter in the next video. Thank you.